Hey everybody, I'm Terri Conneran and I am KRAS Kickers. And I am beyond thrilled today to introduce to you, well, first of all, it's Colorectal Awareness Month. And so happy colon cancer awareness month. And to kick off our KRAS Kickers colon cancer awareness month, we have with us, okay, apologies if I butcher your name because as soon as the camera hits, I mess it up. Dr. Tanios Bekaii-Saab.
Saab,
like the car.
Saab, like the car. Yeah,
Saab,
they do.
like the car.
Links to my Swedish roots, right?
Yes, I should have guessed. And I mean, he is the leader.
It's Arabic. Saab is Lebanese, so.
Okay.
Yeah.
Okay.
And it means difficult. So I'm not that difficult, but that's what the name means.
Okay, well, we're going to hope it's difficult for cancer, right? And so you just
I make cancer's life difficult. That's my goal.
That's exactly what we count on you to do. For sure. Okay, and you are the leader of the GI Cancer Program, director of the Clinical Cancer Research Office for the enterprise-wide Mayo Clinic Cancer Center. And you're with us today. And I had the opportunity to get an introduction to you from the ASCO GI conference that you recently spoke at. And so I'm really happy that you're here with us today.
Yeah. No, it's a pleasure. It's a pleasure to be here. You know, it's an exciting time, frankly, for in GI cancers for targeting KRAS. You know, KRAS is a major driver for our cancer: colon 50%, pancreas 90%, biliary 10 to 15%, gastroesophagus a little bit less than that. But you know, the large ones such as colon, pancreas are primarily driven by KRAS and NRAS, but primarily KRAS.
Now, well, that's a good first start. How does NRAS differ from KRAS?
You know, NRAS is just a different gene that's in the same pathway. So in you know, all this pathway called the MAPK pathway, you know, has these number of genes, protein products that essentially, you think about it from the RAS to the RAF to the ERK, and then to the nucleus. It's almost like, you know, you have that signal that comes from the surface of the cell. Then it's almost like I described it as the Pony Express, right? The Pony Express was never a straight line. It stopped at stations, recharged, and then goes to the next station, sometimes drops a message here and then to the next. You know, this is pretty much what you get. So RAS is one of those first stations. RAF is the next station. ERK is the next, the one down the line. Oh, there's MEK and ERK, and then it sends that signal to the nucleus. When any one of those get disrupted, you know, gets mutated, you start having a signal that is just firing all the time. So instead of having controlled signaling to the core of the cancer cell, you start having this erratic, just telling the cell, you got to keep on dividing, you got to keep on multiplying, and you got to keep on spreading. And so that's pretty much, you know, what happens. Now, KRAS ends up being the most commonly affected gene, and it's one of the first up the line. NRAS is a less common gene, and less effective. Mostly seems to be effective in colon, not as much, say, in pancreas or other cancers we treat. It seems it's a primary driver in colon, but it's only a couple of percent of 2 to 3% of all patients, maybe even less than that. And then you have another one called HRAS, which is incredibly uncommon, and we don't understand it as well. We don't understand whether if it's mutated, if it has the same drive. You know, although they belong to the same family, they're not necessarily as powerful drivers. So KRAS very powerful, NRAS somewhat powerful, and HRAS not so sure.
And when you say powerful, do you mean like harder to target, or do you mean powerful like as far as to the cancer in your body?
Powerful meaning as much as a driver for the cancer aggression. KRAS is definitely a driver of aggression. NRAS likely as well. HRAS, we don't know. We just don't know. I mean, there are studies now looking at trying to shed the only way we know is, I mean, there are two elements that go into this. One is, are these drivers of bad that we can't actually go after? And so that's prognostic, right? That tells us that the cancer is bad, and this is a mutation that drives essentially the cancer to be very aggressive, but it can't do anything about it. So for the longest time, we thought KRAS is just that, right? We never thought we can target KRAS directly, at least. We always thought we can perhaps, manipulate ERK or MEK, these things down the line. But you can imagine when you have nothing is a straight line in cancer. But the closer you get to the surface, the more likely you're hitting a main target that doesn't speak to other targets. The lower you get, it becomes more complicated because then you start having a lot of background noise. So when you hit MEK, for example, which is down the line, you can't stop the cancer, the colorectal cancer cell that has a KRAS mutation from activating. Although it's the same pathway, right? So if you hit KRAS, so if you think KRAS, RAF, MEK, ERK, and you take a colon cancer cell that has a KRAS mutation and you shut down MEK, which is a protein down the line, it doesn't stop the cancer cell from growing. And the reason for this is primarily because MEK speaks to a lot of things on the side. So if you shut MEK, others will pick up. If you hit RAS in colon cancer, you can have some activity. But we'll talk about the problems about just hitting RAS in colon. If you hit it in pancreas, where there is no relevance for an EGFR as much. So if you think about the complexities of cancer overall, you have multiple receptors, and some speak to a lot of different pathways. So we always think, I want to drive from point A to point B. And so if I close down the road, there's no place for that car to go. Well, that would be true if there was the only road to take. But then you have a lot of bifurcations. So you block the road at point B, that car will take sideways. And said that a lot of these genes that are mutated are very difficult to target just on their own, and thinking that shuts down the process. You have to do multiple targeting, and sometimes you have to catch up, and we'll talk about some of the strategies to do with that. You have to catch up with blocking one, then blocking another, then understanding what's driving essentially the aggression of the cancer again, and block that. Now, in some cancers, say like GIST, which is not a subject of discussion here, but say like GIST, gastrointestinal stromal tumors, you find that, for example, having c-KIT mutation, so a single point mutation, and you can hit that mutation, for most patients, you will have an incredible response that lasts for years. You don't need to do anything else. That's not usual. In lung cancer, there are some RAS mutations that are the only drivers for the cancer. You hit them, you shut down the pathway, and the cancer stops dividing. In colon cancer, and we'll get to pancreas, because in colon cancer, it's a bit more complicated. So in colon cancer, you can hit RAS, and then hit MEK, and then hit BRAF. You can hit all these on the MAPK pathway, and you can get a short response, and then the cancer picks back up. Why? So what happens is that, when you block all these anywhere down that pathway, there is what we call adaptive resistance mechanism. And so essentially it's a side road that car essentially escapes all these blockades and just goes back to EGFR and says, hey, now you thought you're not relevant anymore. Now you're relevant again. And EGFR uses a different pathway to send the same signal. It's a little bit more tortuous road, and send that signal to the core. And so in colon cancer, you find that your best strategy is not just to hit KRAS, but also to hit EGFR at the same time. So as you notice with KRAS G12C, which is one of the elements we're attacking right here, that in colon cancer, if you hit G12C on its own, the response rate is 5%, meaning for every 100 patients, only 5 have a response. Why is that? That's because EGFR becomes relevant. And so through some preclinical modeling, as well as understanding, what happens with this pathway, the next generation of trials is hitting now KRAS and EGFR inhibitors with like cetuximab, panitumumab, so Erbitux, Vectibix. And so when you hit the two, then your responses now are 30, 40, and 50%. So you multiply the response by multiple folds. Now that's particular for colon cancer. You can't take that strategy and say, I'm going to do the same for lung cancer, because lung cancer doesn't care as much about, EGFR antibodies and blockade. In pancreas cancer, we had first data was with sotorasib, which is the AMG compound. And in pancreas cancer, and even in biliary cancer, it didn't look interesting at all. The responses were very low. So when we actually, looked at adagrasib, which is the MRTX compound, a little bit more potent, they used a little bit of a higher dose in clinic. What turns out that now you see in the study I presented at ASCO GI, 50% response in pancreas cancer, in which, in pancreas cancer, I can't even get a 50% response rate with chemotherapy, which is amazing. And many of these responses last for month and month and month, and for some, a year plus. Now, again, and pancreas cancer is an incredible, tough nut to crack. I mean, that's a rough cancer. And then there's the thought, do we need to do more than just targeting KRAS? For example, try to go after EGFR, where there are some EGFR relevance in pancreas, not as strong as colon. So the reason why I'm talking about all this is it's very important to keep things in perspective and understanding that just hitting one mutation is not the same across the different cancers. And sometimes we have to be more creative by hitting multiple elements that tend to work together to be able to get the same response. So you can get lucky in lung cancer where you have just one hit, you hit it, and it shuts down. And for some patients, it's almost a cure. And in pancreas, in colon cancer, where one hit is not enough, and you have to do 2 hits, maybe even 3, as we're learning.
I'm listening to you describe everything, and it almost sounds like we're playing like some sort of a complicated soccer match or a football game, right? And some teams, it's like you can attack like this is their weak spot. And other teams, it's like you need to hit that plus this plus that. Am I oversimplifying it?
No, this is absolutely true. It's a strategy. It's all a strategy. And if we want to be too naive about drug development, we just say, okay, I found the mutation, I'm just going to hit that mutation, and it's going to shut down. I don't know if you remember, what was it, 2001? Time magazine had this cover that was talking about Gleevec and CML in chronic leukemia. And I said, we just outsmarted cancer. And the future of oncology, of cancer medicine, is going to be essentially a silver bullet. Well, so it turns out that silver bullet is only for very few cancers. And the way I characterize it more is silver shotgun. You have to hit multiple areas for most cancers, not just one. And colon cancer is the perfect example. Even if you look at another related mutation in colon cancer, which is a BRAF mutation, that's a little bit down the line. If you hit BRAF in melanoma, you get 50% response rate just with one agent. If you hit BRAF in colon cancer, you get 5% response rate. Same thing, because you get that EGFR activated. And when you hit both, you get 30% response rate. Not great, but much better than when with one. And we continue improving that. We see if we move it up the line, it's now closer to 50%. So I think our understanding of the biology of what drives colon cancer and all cancers, frankly, is helping quite a bit refine better our multi-targeted approach. So KRAS G12C, at least in our GI cancer world, is the tip of the iceberg. And when you look at all the KRAS mutations, there are only 2% of all our patients that carry the 12C mutation. So it's the minority of our patients. The majority actually have the G12D, like David, and then V, like Victory. So it's great to see a proof of concept that with G12C, that's the first in class. And I will acknowledge that, about 10 years ago, when we talk about targeting KRAS, it's almost like talking about unicorns. No one would even believe that that's possible. And it's amazing. People will look at you like you have like 3 heads. If you talk about, well, you never know. Like, I used to be lectured by my basic science colleagues. I'm like, listen, great. If you can do it, fantastic, but ain't gonna happen. It's an impossible protein to target. Well, I'm glad that we're proving them wrong, but actually these are the same scientists that had the doubts. But we started with G12C, and it started first, very promising in lung cancer, just fantastic responses. Then in colon cancer, not as good on its own. Then you add the EGFR inhibitor, those responses are looking really good now. Then in pancreas, then in biliary, then in ovarian, then in others. And now we have G12D. And G12D is just entering into the clinic in the phase 1, experimentation. And then after that, I think the next level, I'm hoping that it chose to be, safe enough to move to the next level. And then, that opens up a whole different world. Now imagine 30 to 50% of patients with pancreas cancer have a G12D. KRAS G12C, that's 2.5%. In colon cancer, that's about 40% of all, 40 to 50% of all the RAS mutations, which are about half of the patients with colon cancer. The impact that will have will be just tremendous in those cancers that, I don't know the name yet for the KRAS G12D. It's just an NCTN. It's an NCT number. I see there's a question about this, but I can certainly forward it to Terri.
Yeah, please do, because that is, and I'm a lung cancer patient and I'm a G12D. And so that is probably the question I get asked more commonly every single day is what's going on with G12D. I mean, because there's so many of us, regardless of the cancer location in your body, for sure. Yeah, no,
absolutely. I can tell you the compound's name is MRTX1131.
Okay.
So it's
and is that trial already open?
The trial is either already open or about to open. I think it's only 1 or 2 sites, and I can get you those sites. We have not been at that one site yet. We're hopefully going to be one of the sites in the expansion phase. We will be definitely a site for the expansion phase. I think MD Anderson may be part of the dose escalation, but I'll confirm that, and I'll get you the name of the investigators who are doing this. But the whole area of KRAS. So I'll get you all this information, Terri, if you remind me. But the whole area of targeting KRAS is booming. So I think I don't want to simplify it to just talking about these molecules, these agents that target specifically KRAS. But there are other ways to essentially target KRAS that are still being developed, and I think we still need to also encourage them. One of them, actually, we've led the charge on one of the studies with an agent that's called onvansertib in colon cancer, and now also moving it into pancreas cancer. And just to show you, how these different ways of targeting RAS overall and KRAS specifically are being developed. So in this one agent, and we presented that data at ASCO GI as well with this agent. So it's ONVANSERTIB. And then this particular agent is a polokinase inhibitor. So it's not a direct effect on KRAS, but what it does essentially, it finds the Achilles heel in those cancer cells that have a KRAS mutation, and essentially will make them more likely to be killed by chemotherapy. So it's rather than just shutting down the cancer cell itself, it weakens the cancer cell and makes it more likely to be killed by chemotherapy. And so with this particular agent, irinotecan or formulations that are related to irinotecan are the ones that end up synergizing the most with it. And that study we presented, with others, at ASCO GI, essentially showed in colon cancer in those patients who actually have had, a lot of prior treatments, that there was a response rate of close to 40%. And now this is when you expect, less than 5% response with the chemotherapy only. And some of these responses, I can tell you, they've lasted for a year or two or more. And this is again in patients who have had prior therapy. And we're moving this also in colon cancer, in pancreas cancer, but also continue to expand this. Okay, okay, great. But over 18 months, impressive. I'm not surprised. It works. Is it including chemo or just the agent itself?
Oh, that's not me. Including chemo.
Including chemo. Okay, perfect. Yeah, that makes sense. It appears to need chemo to actually have its best effect.
Okay, so all right. Let me slow you down. You're just like, you're rocking and rolling. I'm loving this.
Okay.
I am. I really am. Okay, so but what you're saying is, I'm not sure about the pronunciation. Onvansertib. Am I saying it right?
Yeah, onvansertib.
Onvansertib.
It took me about a month to be able to pronounce this correctly.
It doesn't come with pronunciation, guys, and I can't even get your name right. And I know how to say your name. Okay.
Hey, listen, so, when you have a name like mine, Bekaii-Saab, things become a little bit easier to pronounce.
So you get a lot of practice. Okay, so let me see if I've got this right. Okay, so it kind of goes in and it just seems to be weakening it so it can be attacked. So it's weakening the cancer mutation so it can attack the target better. Is that kind of what you're saying?
Yes. So the process as we call it is synthetic lethality.
Okay.
So what it does essentially is what a lot of agents we use in clinic, what they do, they induce this likelihood of the cancer cells essentially become relatively weakened and their DNA becomes fragile. And as their DNA becomes fragile, then you hit it with chemotherapy and boom, this is what happens. Now, to Terry's point here, I wouldn't worry about dropping the irinotecan. Once, in my experience, at least once you have a really good response with the chemo plus, then I think it's fair game to let the agent on its own, keep the disease at that level, and then you can always reintroduce the irinotecan if needed. But it seems that you frankly need just a few months of the chemo plus, and then you can go on the agent on its own. So that actually is exciting because these are elements that will expand our capacity to target RAS beyond just the G12C and the G12D until we actually get. Perhaps we're going to get to the day. I should say perhaps. I'm sure we will get to the day, because now we'll see this happening where every KRAS will be targeted, and I'll talk about other mechanisms. Do you want me to answer some of the questions, or
whatever your comfort level is. I mean, you're the star of the show here, so I'm just helping you get to the right and make sure that, yeah.
I want to make sure I get everyone's questions answered. So
go for it.
So this is from Ani. Okay, in the future, can you imagine onvansertib being useful for acquired resistance? It's a fantastic question. You know, the answer is possibly, very possibly. You know, there is no reason, because it's a different mechanism of action. So even if you become resistant to a KRAS, and I'll continue on that front to the next thought I have about where we're going as well with that. But the fact that you're resistant to targeting directly a protein doesn't mean that the protein went away. The protein is still there. And so when you become resistant to it, the KRAS mutation is still there, and its effects, although would find another escape mechanism, right? When you block it, you find another escape mechanism that becomes very difficult to surmount. So for example, you know, as we said in colon cancer, even blocking G12C on its own is not enough. You have to block EGFR and 12C. But then you have PIK3CA, then you have a lot of other, you know, pathways that you may still want to target. The way, you know, onvansertib works essentially is it doesn't directly affect, you know, KRAS per se. It doesn't link to KRAS, but it finds essentially the weak point in those cancer cells that are driven by KRAS. And so if you're resistant to KRAS, that doesn't mean that you're not going to have a response to onvansertib. In fact, it would make a lot of sense to just change the mechanism you're going to target KRAS.
Well, in an oversimplification, it seems like we're getting closer, but you just need to be adding more of the right things. And so it's not going to be just, like they said, a silver bullet. It's going to be a matter of a process and a lot more strategic as far as an ongoing.
Oh, absolutely. Absolutely. And here's the cool thing about all this too. What we're starting to see as well is that, you know, when you start picking on these KRAS, you also irritate that tumor and its environment. It becomes more immunogenic. It becomes more visible to the immune system. And at least in preclinical models, what you're seeing is that you can start combining something that's been done already in lung cancer, immune checkpoint inhibitors, and these G12Cs. And whether that's going to be applicable in pancreas and colon, I don't know yet. We have to look at that. But you know, so then you start becoming smarter. Then you say, you know what? If for 20-30% of my patients, this one agent is doing magic, I still want to see the other 70% benefit from it. What am I missing? And then for those 30%, how can I make sure that their responses are solid and the equivalent of a cure? Because remember, folks, we're all happy with the response, but we all want to see that response that essentially, you know, transforms cancer exactly where we want it to be. It's hypertension, it's diabetes, something that I can treat for life, but you can live with for the rest of your life. Right? And that's how we need to think about the next, you know, the strategy that has been in formation now for years. But now that we're seeing these biologics and immunologics going into the clinic, they're changing completely how we view treatment of cancer. We are indeed smarter, and in an incredible fast, you know, forward time. I mean, this is not happening in 40, 50 years or 100 years. This is every 2-3 years. It's like moving. It's moving. So we love it. I mean, we're just. I think we're seeing. We were seeing the tip of the iceberg. I think we're uncovering the rest of the iceberg now, piece by piece.
Well, it boils down to exactly what you were saying, right? I would rather wear glasses or, you know, have cataract surgery than like be blind, right? And so it's a matter of being able to control it. And even though it's going to keep growing and changing, we're going to just have another type of treatment for it. Now, are we seeing any sort of like co-mutations that are falling into the equation at all?
You know, we're learning a little bit more about these, and there's certainly, you know, elements that that we still don't understand clearly. There are certainly alterations that may make it worse, like PIK3CA, that can certainly, at least in colon cancer, that are major drivers. We have some agents that target PIK3CA. We just haven't seen yet significant activities from targeting them. I think we're getting closer, because I don't think that targeting just one agent is going to be one mutation is going to be enough for colon cancer. And there are others, HER2 mutations, amplifications can be drivers. MET alterations can be drivers. Now, the good news is we have agents actually for all these. The question is, how do we keep up with this, right? So with the advent of liquid biopsies, I think we're transforming how we care for our colon cancer patients and all other cancer patients. So in many ways, and what I do and many colleagues of mine do the same, what we do in clinic is we get our first snapshot at what's going on with the cancer. So let's say, I see my patient first time in clinic. I make sure I have a tissue biopsy. I have a tissue to interrogate, but I also, at the same time, before I start any treatment, I get a liquid biopsy. I get, blood that gets sent for circulating DNA, for, DNA from the cancer cells that's circulating. You remember the days, we're talking about circulating tumor cells and how this is going to change how we do it, but it was very tricky because, the circulating tumor cells are not as stable. They break down, and guess what? When they break down, what do they release? Their DNA. So it turns out that measuring the cell and the cell number is not as good, but actually capturing the breakdown product of the cell, that's the DNA, that actually is good and highly sensitive.
What is in the breakdown product?
So when the cancer cell breaks down, you get DNA, RNA, proteins, and you get a lot of other stuff. The problem is the proteins degrade very quickly. They break down. It's tough to measure them. The RNA that's coming soon with some tests is also easy to break down because they're shorter molecules and they just dissipate faster. But I'll tell you why it's important to have the RNA in just 1 second. The DNA is actually more stable and solid, and so it's easier to measure. So that comes first. The RNA is important because the NTRK fusions, NTRK fusions, for example, these are mostly elements that alterations that you detect through both DNA and RNA. If you don't have the RNA element, you miss on 20 to 25% of the fusions. When you're looking at an incredibly rare target, you don't want to miss on a single %. And so the challenge is, unless you have tissue, and by the way, you have to actually make sure that the tissue is run by a platform that includes both RNA and DNA, because we have common platforms that are used in practice that don't use RNA, so they miss that fusion.
So how would I know if, as a patient, okay, here's a real practical question. As a patient,
So
how am I going to know?
Very easy. I'm going to tell you right now. Foundation Medicine, they don't do the RNA. KRAS, Tempus, Neogenomics, whatever, all these other companies, they do the RNA. I think you have to ask your oncologist and say, do you have the RNA? Is the RNA being tested along with the DNA in that profile? So you as a patient, unfortunately, unless you ask your oncologist, you're not going to know.
Okay, so how would I know, because I know a lot, I talk to a lot of patients, and their doctors a lot of times will tell them, yeah, we did all the testing. Okay, we're being very dismissive with us. And and how do I know that they've done it? Can I get a copy of my report?
You should.
Or what is it that I would be looking for on my report? It's going to say this, so that I know it's been done.
Yeah. So I think it's your medical record. You have every right to have your report delivered to you. Even when the oncologist is reluctant, if they don't want to hand it out to you. So what I do personally, I'd say, I will hand it out to you, but I want to make sure that I spend the time to talk to you about it. because I don't want you to overread into things or miss things that are important. Because, I mean, frankly, even for many oncologists, it's an incredibly difficult report to read. And so I think it's very important to sit down with the patient and go through it. And I do that, most of the times, especially when patients are interested in it. Sometimes patients are not interested. They just, want to put it on you. But I think it's important to see whether you have a DNA and an RNA report. RNA is, it says DNA and it says RNA. So if there's no RNA report, ask your doctor, is there an RNA report? Keep in mind that the liquid biopsies don't report on RNA because they don't have that technology yet. The tissue biopsies do. And when they do it, now there's a caveat because I don't want to oversimplify things. If you think about essentially your internet, your internet power, right? You have just that much bandwidth, let's say 100 Mbps. So when you focus on DNA, you actually get much better results because your bandwidth is 100. That's what you're using for DNA. When you integrate RNA, you still have 100. But now you're splitting a little bit the force. So you may lose on some sensitivities. So I want to put that with a caveat. Now, I don't want patients to be so worried about this because, man, this gets dizzying, right? I mean, what do you think about all this bandwidth? 2 years from now, guess what? We're not going to have that discussion because the bandwidth is going to expand. But right now, I would think that there's very little chance you're going to miss the most important mutations and alterations when you do both blood and tissue. That's why I do both, do blood and tissue. But I want tissue to include RNA because I can't do it with liquid. And if I miss something in tissue for whatever reason, I'm going to find it in blood. Now, and we got to be careful about this. In colon cancer, you're going to find a very high level of concordance between blood and tissue. In pancreas cancer, you're going to miss a lot in blood. Why? Because essentially, think about it this way. What brings on DNA into the blood? It's cancer cells that are multiplying fast and break down, right? The more your cancer is able to break down and release DNA into the bloodstream, the more likely you're going to be able to see that DNA in the bloodstream. With pancreas cancer, it doesn't divide fast and it doesn't break down as fast. And so it sheds very little DNA, so quote unquote sheds very little DNA into the bloodstream. And so we miss about 30%. It's a large number, 30% of those patients. Although with the sensitivity levels continue to improve, I suspect a year from now, it'll be 95% sensitive. So we're getting better every year. But keep that in mind. So I'm telling you this just because there are caveats. It's not as simple as, come on, man. No RNA or, it's more complicated. But I do think that requires a good discussion between the patient and the physician. And I encourage everyone who's a patient, caregiver, to have a frank discussion with the patient. I found it sometimes tragic that patients would go through all the testing. They come to my clinic and I ask them, did you have a test, a genomic test? And they say, well, what is that, doc? And I say, well, did they tell you anything about the RAS, the HER2, did anyone discuss anything to you about the genetics of your cancer? Oh, my genetics are good, doc. I'm like, no, that's not what we're talking about. I'm not talking about your genetics. I'm talking about the genetics of the cancer. And the patient says, no one checked anything. And then I go to the chart and dig deep somewhere, hiding in some parts of the record. There's actually a test there. No one ever discussed with the patient. And it's so embedded in the medical records that the physician didn't even pay attention to it. So not to put burden on the patients and the caregivers, but I think at this point of time, it's very important for every patient and caregivers and advocate to make sure that the patients have this. That's right now, today, I think this has to be the chief complaint on the top line of every medical record is what is that cancer look like from inside?
Oh,
because
totally.
yeah, people have missed on these G12Cs. I see them in clinic. Like, did anyone tell you you potentially could go on this clinical trial? Like, I didn't even know what that means. I get it. Of course you don't know what it means. But did anyone even pay attention to this? You call the docs and you say, do you know what G12C means? No.
It's tragic. It really is tragic. Now, I know it is a lung cancer patient. What is the percentage of people that are not being tested in GI cancers?
In pancreas, it's pretty high. In colon cancer, it's becoming less and less problematic. However, I'll give you a caveat to that. I see patients being tested later rather than earlier for many, which is not good. And oftentimes, if they get tested early, they only get KRAS and BRAF, and that's it. And that's not enough. Okay, MSI high more and more now. But frankly, the first day I see the patient, I want to have all this information on hand. The liquid biopsy, by the way, gives me most of the information I need within 6 days. The tissue may take 2 to 3 weeks. So within 6 days, I already have most of what I need. Now, I understand that a lot of folks say, well, you could wait and it's a resource issue. And I'm like, no, if I was a patient, I don't want to wait. I want to know. And oftentimes your tissue goes and it gets sent back, and I get told, well, there's not enough tissue. Maybe you should try another biopsy, and then we do another biopsy. We've wasted 6 to 8 weeks. We don't even understand yet what's going on with this cancer. And boom, it's BRAF mutate. It has a BRAF mutation. It has a G12C mutation. It has, another alteration. So it's it can be important. But here's another thing for colon cancer. For example, if you have a HER2 amplification, which is about 2 to 3% of patients, I mean, that's a lot, right? You don't respond to EGFR inhibitors. So oftentimes I see these patients coming to me on an EGFR inhibitor, and ultimately, they have a HER2 amplification. And no response, only toxicities, no response. So that's not acceptable practice anymore. Science, some folks with science, would argue that, well, we haven't done the randomized clinical trials yet to say, with HER2 amplifications, EGFR inhibitors don't work. My answer is, you can't do those studies. You can't do a randomized study on everything. And one, we have enough data now from Japan, Italy, and the US that suggest that. Two, we have preclinical data, strong preclinical data that suggests that. Three, other than my personal experience, which is it may be important for some, maybe not for others, but three, there's a biologic rationale. For example, we know that if you target EGFR, so there are three main mechanisms of resistance that happen. One, you phenotypically, right? The genes don't change, but phenotypically, the RAS-driven clones, which are very small percent of clones, become dominant, and the RAS-mutated clones become the dominant, essentially, clones. And so the EGFR inhibition stops working. Two, we see a rise in c-MET. So MET amplifications predict EGFR inhibition. And three, HER2 amplifications, a major mechanism for EGFR resistance. So we know that actually these are drivers for resistance already. So if from the get-go I have them, you're not going to respond to an EGFR inhibitor. And so these are important aspects of how we need to think and strategize in all cancers, but in colon cancer and even pancreas cancer in that sense. So, this is a long way to talk about this, but that goes back to the point and overemphasizing the importance of having a good, I wrote a little paper, in a journal called Oncology a long time ago, 3-4 years ago, and it was, next generation sequencing should be a right, not a privilege.
I 100% agree with you, and I think probably everybody here does as well. And I don't think it's just a matter of having it done. I think it's finding out what the results are, because as you know, as a doctor, it tells you what best treatments are for me or what is not a good idea for me. And as a patient, it's empowering to know where it is I need to be looking for information and what type of data it is I'm looking for. You know, you don't know you need a I mean, you don't know you need a new car if you're looking for a vacuum cleaner, right? So it just helps to narrow down the focus.
Well, you know, back in the days, a doctor couldn't essentially take care of patients without a stethoscope.
Mm-hmm.
You know, a couple of years ago, you couldn't take care of a patient without a scan, without knowing what's going on. Today, I think it's unethical, and that's my viewpoint. People may argue with that, but I think it's unethical. It's impossible to work with a cancer patient without having a genomic profile. Actually, two things. You know, we just published a paper in JAMA Oncology that suggested that we miss 30 to 40% of patients who have inherited cancer. We actually miss on these genes if we just went by family history alone. Because we took 3,000 patients with all types of cancers, mostly GI, it ended up being mostly GI. But just talking about the GI cancers at least, what we found consistently is that if you just sit down and follow the usual algorithm of, you know, tell me about your family history, the types of cancer, etc., you miss about 1/3 of the patients. You know, the implications for that are huge.
We're sitting right here. We know the implications.
Exactly. They're huge, right? They're huge. And I think we're only shaving the tip of the iceberg with this. So we learned quite a bit about a lot of, you know, variants of unknown significance in patients who actually have family history. And we're trying to compile those and see if we can understand clearly whether those are also driver genes.
What are the primary genomics are different than genetics, just to clarify for the folks. Okay, but what are the primary genetics that one inherits that leads towards any type of a colon or pancreatic cancer?
Well, Lynch remains number one. BRCA1. So the usual offenders are there, right? Lynch, BRCA1, BRCA2, PALB2, but then you have MUTH, you have ATM. You know, a few patients with ATM mutations that either develop pancreas or colon, and these are, you know, genetic inherited germline, and that has significant implications. NBN, so N like Nancy, B like boy, N like Nancy, and these typically, you know, they may have capacity to be vulnerable to synthetic lethality compounds. So agents like PARP inhibitors or even onvansertib. So you know, I had a couple of patients with germline KRAS mutations.
Really? Mm-hmm. In colon cancers.
Colon and lung. So they had both. They had both. It was I've never seen it. First patient I've seen was like, I've never seen that. So I actually, you know, called the company, said, "Are you sure?" I said, "Yeah, I have never seen it in colon, at least." But they exist, right? And we're learning more and more about them. So every patient comes to my clinic, they get the genetic profiling, the genomic profiling, tissue, the liquid, and depending, you know, if it makes sense or not, they get also that MRD assessment. For most patients, it doesn't make sense. For some, it does. And so we have this whole profile. So I'm looking at this longitudinally, you know, I follow with blood, especially at the time of progression, you know, at multiple points. We have a large platform called Colomate, which is, you know, essentially a platform that's the largest platform in the United States that essentially is going through our research consortium. I can send you more information, Terri, about this. But that platform essentially has a point of entry for patients who failed other lines of therapy. We do liquid biopsies on all patients, and then we have multiple baskets that patients get assigned to based on their genotype. And then we follow patients with essentially what we call liquid biopsies, so we understand what is driving the resistance, why is the patient less likely to respond, etc. We are working with our Japanese colleagues as well. You know, they have another huge platform called, believe it or not, Godzilla. uh, but we work very closely with them. And what we're doing now, we combine what we call the rare target initiative in colon cancer and biliary cancer and pancreas cancer. So we're all GI oncologists. And so what we're looking at is essentially these unusual BRAF alterations, the unusual RAS alterations, not the G12C, the less common ones. Also, we look at allelic frequency, the low yield versus the higher yield. So they have thousands of patients that they've collected data on. We have, 1,000 plus, and we'll have about 5,000 by then. Imagine now we'll bring the power of those two different patient populations treated, somewhat similarly and others differently. So we have, I tell you, a team of like 20 clinical researchers from every part of the United States, every part of Japan, working together on this initiative. We're gonna present some data already at ASCO, I hope, if ASCO allows us to, but also ESMO GI. So every few months, we're gonna start releasing more data. And that data essentially will become more public and hopefully will stimulate more research. So a lot of things, we wanna do and others wanna do, and these collaborative efforts, I think will continue transforming the field. Maybe time to take some questions if you want me to.
No, for sure. But I do want to say, please make sure that when if any of your abstracts or anything you're going to be presenting, please let me know. Drop me an email,
Okay.
send it in, because that's part of the information that we're really desperately trying to glean. Okay. And that's really what makes the difference, because as you've already pointed out a couple of different ways, the patient is still the one that has the most vested in this.
Yes.
And we're the ones that stand to gain or lose the most. So, like it or not, if we don't advocate for ourselves,
well, I'm with you.
Yeah,
I'm with you. Knowledge is power.
it is totally power. And I'd rather take something to my oncologist and have my oncologist say, well, that's not going to work for you, than not even ask the question. Because,
So
like I said, go ahead.
Let me tell you something here, because I think it's important to empower patients. I mean, it's not being antagonistic to oncologists. And the way I train my fellows, I say, it's very important to never take it personally when a patient comes and challenges you a little bit. And they're not challenging you because they think you're an idiot. They're challenging you because it's their life. And they want to know, and they want to have that information. And you actually have to walk them through it rather than to take it personally, because, some doctors, no offense, tend to take things a little bit more personally than they should. And they think you're challenging them, meaning you're questioning their expertise. You're not. And maybe, some in an introductory way, hey doc, I'm not trying to challenge your I'm here, my life is in your hands, but I would like to know a little bit more about this.
Oh, completely and totally.
And if they say, let me tell you what, if they say no, and they say, no, this is not a point of discussion. If you don't like it, just go somewhere else. Go somewhere else.
I totally agree with you. Completely and totally agree with you. Because
Yeah.
when we walk in, and it doesn't matter how fantastic of a doctor you are, right? How world renowned, how much research you've done. I'm 100% vested in this as a patient, right? And I have to trust you to choose the right toxic chemicals, to do the right sort of surgery, the right type of radiation, or whatever it is. And I need to have that kind of confidence, because as a patient, I'm going to have more success just having the confidence that we're doing the right thing in general. So how do you feel, or how do fellows feel like when we ask for a second opinion?
You know what? See, whenever a patient asks if they can have a second opinion, which, for me, as I get more gray hair, it becomes less and less something that my patients ask. Plus also I'm super specialized, but still you get the occasion. And I make it very clear that I do encourage a second opinion if the patient feels that they need it. And the patient, that's what my fellows hear. And they say, if I tell you, this is how I address with it. If I tell you, you can't have a second opinion and come back to me, you shouldn't be my patient. It's okay. Now, here's the other thing I would say though, as you know, as an expert in the field, if you want a second opinion, please help me, let me help you find the right person to talk to. Because oftentimes what I see is my patients, previously, my patients would actually go and get a second opinion from some shop down the street, from someone who has no clue. Maybe has a clue about oncology in general, but is not a super specialized person. How does that add value to the patient? That doesn't add value. So when I want to add value, I want to get to someone who actually specializes in the field. So I would say that it's very important, to have a frank discussion with a doc and ask the doc who is a trusted person they can be referred to, rather than come and say, I want to go see Dr. XYZ, if you don't mind.
So you're saying, okay, so like if I had a car and I just took my car to the shop, it's completely if I'm taking a Ferrari to a general car dealer, it's completely different than going to the Ferrari dealer. And each one of us are our own Ferrari. So we need to make sure that we at least get to see one top tier doctor, one top research that's very specialized in what type of cancer that we've got,
Yes.
so that we know what we've got going on.
Yes. So so let me tell you how I practice. So the first day I meet my patients, we go through quite a bit. We talk about everything. We also talk about the importance of our partnership. We're partners in this. Yeah, as you said, you have vested interest, but I have vested interest in making sure that you're gonna do great. And my goal in treating you are twofold. One, focus on quantity, but also focus primarily on quality. Quality and quantity go hand in hand. If I'm going to sacrifice quality for quantity, I'm not doing a good job. If I actually sacrifice quantity, even if I give you quality, but also I have to be careful, because some folks care quite a bit about quantity. Some folks don't care as much about quantity because they're at different stages of life. We'll all go through this. So we have to have a frank discussion. What is it you want? What are your goals? Because the goals of the patient are the most important goals at the end of the day, because they help me essentially establish a strategy. There's not one patient that's the same as the next one. Absolutely not. When we talk about individualized care, other than the genetics and the genomics and all this other stuff, the first individualization is individualization of goals. Every patient has a completely different set of goals. Some are a little close and some others very divergent, and we have to respect that. And you start with this, because I feel oftentimes that patients come in and they're almost apologetic about trying to say what their goals are, because they feel like they're gonna piss off this guy sitting there who's a world expert on cancer, and now I'm gonna tell them what I think they should do. No, you know what? It's a partnership. But the other thing is also, part of the partnership is to always have touch points, understand more. I have a team, I have nurse practitioners, a lot of folks that work with us. So I'm not always there, but I'm always there. I'm not always physically there to see the patient every single time, but I'm always there, meaning that nothing gets taken care of unless I give the checkbox. But that said, the whole team works in the same way I work. We have the same approach and all this, and that's important, for our patients. I mean, you are part of that decision, whatever that decision is, from day one, and we don't want to take that autonomy. That's patient autonomy. That's the first principle of medicine, right? Do no harm and autonomy of patient. So do no harm can mean a lot of different things. I can do harm but with all the good intentions. Okay. I mean, I can still hurt a patient by thinking I'm doing the right thing. So I have to be careful. It could be financial. It could be, through giving a drug at a higher dose or a lower dose, whatever, all these things. But the principle of autonomy is actually, I think, the most important principle in medicine. That's a respect. That's a respect of the human being that's sitting in front of me. And we're talking about essentially a disease that may take that patient's life away. I mean, nothing can be more profound than this. And that autonomy is the most important driver of how we take these decisions in a way that fits the goals of the patient. So that's apart from all these discussions about KRAS mutations and all. These are important, but I think this is something that if we don't address from day one, then we haven't gained the full trust of our patients.
But you and I keep coming back to the same place. We as the patient need to trust the doctor.
Yes.
But we also need to be able to be willing to open our mouths, have
Yes.
that conversation, have that dialogue, and say, you know, this is really important to me. I really want to live to be 120.
Yeah.
And now you may have to like scale that back a little bit, right? But at the same time, we need to be able to be clear and have that open dialogue, and having these conversations that we bring in here in KRAS Kickers, it's like it gives us that sort of permission to advocate for ourselves and tells us that it's all right for us. If you this Ferrari doctor, right? Okay, this top tier expert doctor in the field, okay, is telling us to do this, then that means I should do that, and I can do that as a patient.
Absolutely, absolutely, and you know, I get challenged by my patients, like everyone does, and actually I love it. And oftentimes it leads me to you know, maneuver around or change course a little bit. You know, I can't tell you about the moments where the patient says, well, doc, what do you think? Is this the right time to do this or to stop here? And I'm like, you know, that's a great point. I think it's reasonable, but let's talk about what that means. So being challenged is not necessarily that, well, doc, you know, I don't trust you, man. Why are you continuing this? No, it's important to have that, and I think, you know, it's important to have that partnership, man. I mean, especially these days, it's medicine is so complex, and our patients, you know, patients overall with cancer are doing fantastic. We're living the longest ever, and God willing, you know, we're going to get to that cure at some point. We're getting already to a cure, by the way, and even stage 4 cancers, and many stage 4 cancers, including the ones I treat, we're getting to a cure. Okay, stage 4 being cured. So stage 4 is not necessarily for every single patient the end. We are curing many stage 4 cancers, not all, not most, but many. And so I want to make sure that it's clear that oftentimes the family physician sends a patient and says, you have stage 4 cancer, you know, go pack your stuff, and you got 6 months and you're done. And then, you know, 5 years after, I asked this patient, go back to your primary care physician. Maybe, you know, there'll be a teaching moment not to say that to the next patient, please. So anyways, let's take some questions.
Okay, for sure. For sure. Okay, now I've seen the cholangiocarcinoma question about the G12.
Yeah, I'm gonna, so I see that there was, starting with Jill Rupp, that's at 115, maybe, that I work in an academic setting. Do you think community oncologists are testing for KRAS G12C? The answer is any test they sent, if they send a next generation sequencing test, whether it's Foundation Medicine, it's KRAS, it's Tempus, it's NeoGenomics, and I'm not advertising for any of them, there may be others. Whatever test they send, as long as it's a comprehensive test, they should include KRAS G12C. So that's reported out. So like Terry was saying, you have the right to have access to the test results, and you look at it. And I encourage you to do that, because I've seen a lot of patients that have no clue, not because of their fault, but because nothing was ever shared with them. Ask. Ralph, any thoughts about KRAS? NRAS G12C, I don't think that I've seen any of this. But KRAS G12C in cholangiocarcinoma, you know, we actually, part of that study we presented at ASCO GI had about 8 patients with cholangiocarcinoma, and 50% of them had a response. all of them essentially had disease control. And actually, they did even better than pancreas cancer. Now, the caveat with that is that it's only 1% or less. So it's even a more rare. There's it's even less common than in pancreas cancer. But if you find it, yes, you target it with adagrasib. So sotorasib didn't do as well. Adagrasib, the agent, MRTX agent, seemed to actually have pulled an equal likelihood of a response with KRAS G12C in cholangiocarcinoma. So the answer is same excitement that I see in pancreas, I see in cholangiocarcinoma when you have the KRAS G12C.
I love doctors saying that they're excited, because that's only good things. Those are happy patients that get to attend weddings and go do fun things.
Yes, absolutely. And I'll tell you, when we saw the data with adagrasib before I presented it, I'm like, man, thank God, because I had my doubts since sotorasib didn't look as good. I mean, it looks equally good in lung and somewhat good in colon, although I still think there's a little bit of an edge to adagrasib. But I didn't think it's going to be that big between the two when it comes to pancreas and biliary. It's pretty impressive. Hopefully, these will continue moving forward. Yeah, the force of Godzilla. Yeah, it is. Our Japanese colleagues are just amazing with their with the way they find these acronyms. But of course, Godzilla is a force in Japan. But I have to tell you something. Our Japanese colleagues and investigators are just amazing. I mean, they do things that I don't think we have the capacity to do in the Western world. They're so organized, so clean with their data. But here's the beautiful thing about their platform, and actually we learned quite a bit of that from them. Their willingness to share data is amazing. They're not like sitting on this data and they want to hide it so they can publish, and they are willing to use it, and so they care about this. And so we used our Colomate platform the same way. So you know what? More power to them, and more power to anyone who wants to collaborate. We're willing to share that data as long as that data is shared for good use. So they're very open platform, and we want to make Colomate ultimately an open platform, as we should, because ultimately I'm not doing it so I can get another paper. I don't need another paper, but even if I did, that wouldn't be serving my patients. We want to do it for for drug development and for essentially ensuring that our patients continue to do well.
I gotta say, Dr. Saab, we really appreciate your heart. It's very clear your passion and your heart for your patients, and for us, not just as patients, but as people. So thank you for what you're doing and being such like a leading voice in it.
Well, it's my pleasure. I say, the best part of what I do in life is, and I do a lot of things, is actually being in clinic and being with patients. And I didn't take this on. One day I'll tell you the history of my family. It comes from a lot of physicians on my father's side. My father is a lawyer, but one of the two striking figures in my life were actually two uncles of my father. One on his maternal side, who essentially was in the mountains in the Bekaii Valley, who used to, on a horseback, there weren't roads, on a horseback, go from village to village. For a whole month, disappears from home and comes back. And he was a general doc and would treat everything, and would come back with the chicken and tomatoes and whatever, And he was from a comfortable family, so he didn't care as much about him, but never got paid. The other one was actually also a family physician who, my sister had asthma. We were back when I lived in Beirut. She never went to an emergency room one day of her life. When she was a kid, she'd have these attacks at midnight. He would come sit by her side with nebulizers, 3-4 hours, steroids. till 4 a.m. and then next day would go back and go to his clinic. And so, this is just to give you an idea about, the folks I got exposed to growing up, and I think they were great influence and why I chose medicine. Anyways, I'll have more stories.
Yeah,
I'll have more stories.
So the part of it is genetic for you, and part of it is like with the modeling. But I mean, it's very clear. I mean, I don't know you really well, right? But I mean, I can see and we can feel your passion, and so I really appreciate your being able to share that passion and giving us that power as a patient. So thank you for doing this. And I'll get, yes, I can tell I'm getting you embarrassed. And so I'm going to move back to the questions about CAR T therapy, because I nothing worse than embarrassing my guests.
Yeah. Well, so CAR T cell therapy is tricky in colon cancer. So far, we haven't seen the same signals, that we've seen in diseases that are typically immune driven. So think about it like this. I mean, CAR T therapy is immune therapy essentially. It's immune therapy times 10. And unless you have an immune milieu that is conducive to responding nicely to immune stimulation, then you're not going to see the same levels of response. So the good news is that KRAS mutated colon cancer can be stimulated to move from this cold front to this warmer front. The bad news today, at least, but I'm hopeful that there will not be a problem in the future, is that we can do this. It's gonna just take a clear understanding. We've already failed a few steps, but failure is not failure. Just to be clear. Anytime you say, another negative trial, but another negative trial will mean that that's one checkbox that I don't have to waste any more time on, but I understand more about the biology. And it's almost like, you're coming from multiple fronts on the battleground, and you may lose a battle here and there, but then you start winning other battles elsewhere, and then you win the war. So I think losing battles is only empowering us to get closer to winning the war. So there's a lot we understand about the negatives that help us understand the biology better. So we're not there, we're getting closer. There's a question about, I think on top from Maureen, about A1460, KRAS P10, ATM, driving force, anything new. So ATM can be a driving force, P10 not as much. KRAS is definitely driving force. The A1460, is that the KRAS mutation? Or is that a mutation standalone? I don't know what that is.
I'm assuming she means that it's a standalone based on
KRAS A, yeah, KRAS A maybe. I should probably put that in somewhere down there, and then I can comment on it. And then if someone has a KRAS G12D and an ATM mutation.
Okay, let me read it out because let me read it out loud because we're recording this, so it'll make more sense.
Oh,
okay?
I'm sorry.
So
someone...
let me feel like I'm participating too. Okay.
Yeah.
If someone has a KRAS G12D mutation and an ATM mutation, and a trial that targets the ATM is available, is it worthwhile to target the ATM or hold off for a trial that targets the KRAS mutation? That's part one.
Yeah, no, so so it's difficult to say which one is the major driver. So they probably are both relative drivers. Here's the good news though. So, there is data that suggests that a platinum or even a topoisomerase inhibitor, such as irinotecan, can make those cancer cells, ATM mutation will make those cancer cells vulnerable to the effects of irinotecan. So an ATM mutation and a KRAS mutation would, if that's colon cancer, would be a good, that patient would be a good candidate for an irinotecan plus onvansertib, because you're hitting both. You're hitting the ATM, you're hitting also the KRAS. But if you had to prioritize, it frankly will all depend on what you have available. KRAS I think, is probably a bigger driver than ATM. Ideally, you want to try to see if you can hit both at the same time.
Okay, so let me recap. So ATM is inherited from your parents or someplace in your lineage, correct?
It's both. So
It's
you
both.
have the somatic and the germline. Yeah, it can be just somatic. I mean, we've seen both. There is no real difference in how they respond to treatment. You know, it's
But
just,
if somebody has an ATM gene, or ATM mutation, does it typically respond better to a platinum-based drug? Is that what you're saying?
so we know they respond well to platinum. Or there's emerging data that also they respond well to topoisomerase inhibitor. In my experience, they both work equally well with ATM. And so the short answer is, for colon cancer, you know, and even for pancreas cancer, the typical chemotherapy regimens we have work pretty well with ATM mutations.
Okay.
In fact, you know, we presented some data at ASCO GI from a study we had with nanoliposomal irinotecan and a PARP inhibitor. And we had many ATM mutations in there. You know, I'll send you that poster as well. And that essentially shows that some of these patients do extremely well. It's a small number, but they do extremely well with that targeted. So we do have strategies being developed, certainly, that includes those ATM mutations.
Okay, that's good to know. So it is starting to boil down into the co-mutations. It's not just the one thing, it's the combinations. And it speaks to what you were saying about the individualized treatment. I mean, we're the person in the personalized medicine. And so we need to make sure that we're taking the whole person into consideration.
Exactly.
Okay. All right. I had a question sent to me about tumor infiltrating lymphocytes or immunotherapy with KRAS mutated colon cancer.
Yes, I think that also relates to the CAR T cell question, but I'd say that, you know, in KRAS mutated colon cancer, there is not as much tumor infiltrating lymphocytes or TILs, unless there is a microsatellite instability high phenotype or some high tumor mutational burden. Typically not. The KRAS G12C, excuse me, the KRAS G12C mutated colon cancer may have a little bit more of an infiltrate, but I think again, you know, at this point of time, most of these tumors tend to be relatively cold, absent the presence of an MSI high or a high tumor or Pol E, Pol D. So these can also significantly elevate the tumor infiltration lymphocytes. That's about 3 to 4% of all patients with colon cancer, regardless of KRAS status.
Okay, let's see, I'm going down. There's 2 ongoing trials with KRAS G12C and the parameters. So they'd like you to address those two, address that.
Yeah, no, so there are multiple.
Yes.
But I can speak to the Mirati studies and somewhat to the Amgen studies. But there are also others, by the way. I mean, there are Novartis, Lilly, you name it. Everyone wants to jump into the
Genentech has
G12C
one.
space.
Yeah, Johnson has some. Yeah.
It gets dizzying. I mean, I think ultimately, you know,
Thankfully, it gets dizzying. And shout out, just so you, in case you don't know this too, and anybody on the call happens to be new to the KRAS Kickers folks. Okay, if you go to the KRASkickers.org, there is a tab there where you can download all the KRAS clinical trials. And we're gonna be circling back with you to get the information that you've got so that we can keep this information dated and fresh too, because that's really what it's all about. So okay, back to yeah.
Excellent.
Okay.
So I mean, I think, and I may appear biased when I say that, but I think so far adagrasib is the one that impressed me the most. You know, but you know, it doesn't have an approval, so it remains on clinical trial so far. But at least in those cancers that are very sensitive, like lung cancer, and colon cancer when you combine with EGFR inhibitors. I think sotorasib and adagrasib do about the same. But when you start looking at other cancers that are a little bit more resistant, I think adagrasib is getting the edge, historically at least. Now, the two trials that are ongoing, at least with adagrasib, major trials, and one is the basket trial that still has, G12C arms, single agent, adagrasib plus minus cetuximab for colon, for patients who failed multiple lines of therapy, for pancreas cancer, for biliary cancer, for ovarian cancer. They also have lung cancer in combination with a number of different combinations with immune therapy, with pemetrexed. There is ovarian, and so it's really one big trial, basket trial that has a lot of arms. And the other trial, that's Crystal 10, that actually is a second line trial. Patients have to fail first line FOLFOX in colon, and then their second line is FOLFIRI plus minus adagrasib. So you get, go ahead, please. No,
that's okay. I just know that you're tight on time today too, and I had somebody I had promised a question to, so I want to make sure that I get it asked. So I want to make sure you feel like you answered it though.
So just quickly though, can I say, when someone says when is the optimal timing to do a clinical trial, I said all the time.
Okay.
If a clinical trial is available, that should take priority. This is how we get these life-saving agents into the clinic. And frankly, this is when we talk about partnership, this goes beyond partnership. And I remind everyone that the most human act is when patients volunteer on clinical trials, because they're volunteering to uncertainty when they do that, right? We're always talking about some level of uncertainty, added toxicity, maybe not added much benefit. And without essentially volunteering, that's a selfless act. There is some selfishness to it, and that's okay. Nothing wrong with that. We all have selfishness, and selfishness is not a bad trait, right? I mean, we're selfish because we want to be with kids, with parents, with loved ones, with friends, whatever, right? But there's also an act of selflessness, a big act of selflessness, and that cannot be undermined. But I would say that whenever there's a clinical trial, it should be your primary option when it makes sense.
And let me say that, because I didn't come from a medical background at all or a science background at all. I thought it was just like, as a patient, I just showed up one day and say, okay, that's it, I'm volunteering. That's not how that works. They qualify you to make sure that you're appropriate for the clinical trial.
Yes.
And so it's not like literally grabbing people off the streets and just dumping drugs into their bodies. They're making sure that you're a good match for it, and they're comparing you to a standard of care. And the reason I point that out is because I just didn't know better. So, okay, so here's the question I promised somebody. Okay, is they wanted to know about KRAS G12D, which I know is a small group. Mm-hmm. And can you just give us some information on it?
Yeah, so G13D mutation, as you said, is one of the primary drivers of KRAS. And so in pancreas cancer, it's half of the patients with pancreas cancer. In colon cancer, is about 20-25% of all patients. It's actually the majority of the RAS mutations. Across the board, it's the primary mutation in GI cancers. So it has the same relevance as G12C. And as I said, it's the mutation that now is finally has an agent that is likely to target it. And
G13 has an agent that's going to target it. What agent is that?
G12D. Is it G12D or G13? Oh, I'm sorry, G13D.
1-3.
Oh,
no, that's
okay.
No,
I've already talked about G12. No. I'm sorry.
No. That's probably maybe I misspoke too.
No, you did not. You did not. Okay. My mind is I think my mind is fixated on that G12D.
Yeah.
So G13D.
I'm hopeful.
G13D is a less common alteration in colon cancer, and G13D is a very aggressive mutation. It's probably one of the more aggressive KRAS mutations. There was a time when we thought G13D mutations are not necessarily mutations that drive resistance to EGFR inhibitors. And there was a large study in Europe ultimately that showed us that that's not true, that it's a driver of resistance to EGFR inhibitors. So today there are no agents that directly target G13D mutations. And I don't think, at least in the foreseeable short term, we're gonna see that happening. Maybe, again, as I said, a few years from now. Now, that doesn't mean that agents, such as, onvansertib, would not have the activity there, right? Because we're using a different mechanism, not a direct mechanism, but we're taking advantage of its presence. So some of the vaccines that are being developed, some of the siRNA strategies that are being directed, more personalized, quote unquote personalized, alterations. So overall, right now there's no direct targeting of this mutation.
So it's just kind of standard of care for right now, and like anything else, we just kind of keep taking it down the road as long as we can.
Yeah, absolutely. Yeah. And agents that indirectly target are very reasonable.
Okay. And are there pan-KRAS drugs that are working in colorectal?
Like targeting all KRAS, no. Yeah. I mean, nothing that directly target all RAS, no. And that would not be possible, frankly, in a way that would be efficient, because you really need to lock into each one of them in a very specific way. They're very difficult, proteins to target.
Okay. All right. And then last question, because like I said, I'm trying to be respectful of your time, and we have a tendency to run over. So I'm trying to stick with it. Okay. How many months off between KRAS targeted therapies is necessary or encouraged for acquired resistance? What would you suggest?
It depends. It depends. So I'm thinking about just the frame of the question. So between one and the other. So if we're targeting, let's say, to go back to the discussion, we're targeting KRAS G12C, and you become resistant to KRAS G12C inhibitors, then I think within 3 to 4 weeks you could go to onvansertib, which is a non-specific way to target KRAS, because the mechanisms are not the same. Now, there are KRAS G12C inhibitors, such as, for example, the Lilly compound, which is being developed in both patients who have been exposed to other G12C inhibitors and those who have not. And the thought is that it links differently to the target. Now, again, I haven't seen data to suggest any other way, but there's really no, at this point of time, no good time to think that acquired resistance can be reversed. I'll say one word about this. The whole concept of re-challenging patients with, say, EGFR inhibitors is something that exactly relies on that principle. So when you block EGFR, ultimately you get these RAS clones showing up. So you can't target with EGFR anymore. You use chemotherapy, you clear those clones, you recheck with the blood, and what you can see is that essentially your RAS has become to a certain level again. The primary driver in 20–30% of the patients would be those clones that essentially are KRAS wild type, and you go back with the EGFR inhibitor. Just what I mean by that is that oftentimes you may need to go to a non-specific strategy first, let's say, after resistance, to clear those clones that are the most resistant with chemotherapy, the cluster bomb, and then you go back to a targeted strategy.
And about how long would you put between those if you're just kind of like,
so it's 3 to 4 months ideally is when you see those trends changing. Too early may be a little bit risky, because then you may not actually uncover for all patients. Now you can do it, right? That doesn't mean you can't do it, but you may not uncover those specific patients who actually are the most likely to do best. So this is where the principles of liquid biopsy, serial liquid biopsies become very important. That's why we do them in my clinic and other large academic centers, because we have these trials lined up for our patients, right? I mean, you know, I'm waiting around the corner with a liquid biopsy. If I find something and I have a target for it, I'm going Zoom to it.
Totally. Okay. It's 3 or 4 o'clock. So like I said, I have a tendency to run over. So I must really say thank you so much for joining us today. You've been an absolute pleasure to have in, and it's been so helpful and so informative for everybody to have you here. And I really appreciate everything that you did and everything that you're doing and you're continuing to do. So please, I look forward to getting that information from you. And I'm hopeful that if you're going to be, I'm planning on being at AACR and ASCO. And so if you're at those places, well,
ASCO for sure.
Yeah.
ASCO for sure. And let's set a time from now if you want. Unfortunately, AACR didn't work out as well. I just had other meetings.
Well, you're a busy guy. It's okay.
Yeah.
So yeah, we'll have you come back after ASCO, because I'm sure that there's going to be some very interesting data that should be released, and it should be pretty cool to be able to do that. And if there's anything that's going on at that time, maybe we'll be able to do some stuff and do like a quick little drop down and do live sort of a video there on site. That'd be very cool. So thank you so much for doing it. And I'm looking forward to getting that information back from you. And you guys, thank you. And thank you all for being here today, because, you know what, we're all learning as we go. And, you know, it's really been just an absolute pleasure to hear how important it is for us to stand up for ourselves. And, you know, we had our Ferrari doctor tell us that our bodies need to be taken care of, and you're entitled to it. So thank you guys all for joining us, and we'll catch you all next time. Okay, thank you so much. We'll take care. Okay, bye-bye.
All right, bye-bye.